Thank you for inviting me to share our recent work on testing PFAS in globally sourced food packaging using targeted and non-targeted approaches. Because of PFAS' widespread use and persistence, they have become ubiquitous contaminants in the environment, food, and humans. Diet is considered one of the major sources of PFAS exposure, and their use in food contact materials, along with their migration into foods, can be a potential source of this exposure. Indeed, recent research demonstrated troubling correlation between consumption of packaged and prepared foods and human body burdens of PFAS. This study was published in Environmental Health Perspectives in 2019. The scientists analyzed PFAS serum concentrations in the U.S. population collected in 2003 through 2014. And correlated PFAS data in serum with reports on dietary intake from the participants. What they found was consumption of meals from fast food and other restaurants was associated with higher serum of PFAS concentrations. However, inverse associations were noted between serum PFAS and food eaten at home, which was purchased from grocery stores and wasn't packaged. These findings led us to the goal of our study, to test PFAS in consumers' food packaging available on the U.S. market to better understand the role of diet in PFAS exposure. Generally, chemicals found in food contact materials can be either intentionally or non-intentionally added substances. What is interesting and unique about PFAS in food contact materials is that they can represent both categories. Intentionally added substances are chemicals purposely added during the manufacturing of food contact materials, and PFAS are added for water and grease resistance. Non-intentionally added substances are products of degradation, reaction products, results of contamination, and we will show you in non-targeted part of this presentation that some PFAS intentionally added were degrading into non-intentionally added substances, and also food contact materials can be contaminated with PFAS during production and through recycling processes. Selecting food packaging samples for the study, we aim to cover samples from different geographical origins, representing different food types and different consumer groups, with the ultimate goal to estimate the exposure. With this in mind, 88 food packaging samples were collected from 13 supermarkets in Pennsylvania, USA in 2021. After acquiring packaged food and recording information on geographical origins and packaging composition, food packaging was separated from the food, rinsed with water to remove particulates, and stored in individual plastic storage bags. As you can see from this diagram, the samples originated from the USA and 23 other countries and areas. In terms of food types, the samples were separated into bakery, dairy, meals, produce, dry meats, and miscellaneous others, mostly snack categories. Here are some pictures of the food packaging we tested. The first column represents dairy with cheese and butter. Here you can see bakery types with cookies, pancakes, some examples of meals and produce, dry meats, and the category we call others, which is mostly snack, popcorn, chips, animal crackers. This is all common food you see in grocery stores. For sample preparation and analysis of food packaging samples, our goal was to get as much information as possible to widen the analytical scope and to provide a better picture of PFAS levels and food packaging samples. For sample preparation, we used several approaches. First one, extraction, to answer the questions, what PFAS are in the samples and measure their levels. What PFAS are we not capturing using targeted analysis? For this, we use total oxidizable precursor assay, TOP, to measure PFAS precursors. Finally, to assess a realistic picture of potential PFAS migration into foods, we used migration testing performed under controlled conditions. For analysis, we use targeted and non-targeted approaches. Here in this presentation, I will mostly focus on targeted analysis, and my colleague Kevin Strosky will cover non-targeted analysis of these samples in the following presentation. Here's the list of our 34 analytes selected for targeted analysis. They included the carboxylates and sulfonates with various chain lengths. Carboxylates were C4 to C14, sulfonates C4 to C10. Also new and emergent fluorinated ether compounds and other telomer sulfonates and sulfonamides. We also included 20 isotopically labeled internal standards. PFAS analysis was performed using a UHPLC mass method with a Waters LC outfitted with a PFAS kit to replace all Teflon tubing with PEEK and include a delay column. Our total runtime was 15 minutes with a 5 microliter injection time. The LC system was connected to SCIEX 6500 Q-trap via contact closure, and here are the conditions for both LC and MS. As I mentioned, we had 34 PFAS analytes for targeted analysis plus 20 isotopically labeled internal standards. Calibration curves were constructed ranging from 0.05 to 5 nanogram per milliliters. And here I listed our LODs and LOQs for PFAS in this method. We also performed non-targeted analysis with high-resolution mass spectrometry, where we connected the same Waters LC system through a contact closure to Q-exactive Orbitrap mass spectrometer from Thermo Fisher Scientific. This chromatogram shows separation of 34 targeted PFAS and 20 internal standards within 15 minutes chromatographic run at a concentration of 0.5 nanogram per milliliter. This is a middle point of our calibration curve. You can see that we were able to separate them quite nicely from 2 minutes to 9 minutes. However, as we all know, PFAS analysis can be challenging, and I want to point out two problems that we encountered. Here's a chromatogram again showing separation on SCIEX 6500. And the first problem we had was high PFBA background contamination when we used MRM transition 213 to 169. So it showed it in all of our samples. And at the bottom chromatogram, you can see PFBA peak showing for this transition in solvent injection. Since PFBA only has one transition using triple quadrupole, US FDA recommends confirming it with high-resolution mass spectrometry. This chromatogram on the top showing separation of the same PFAS standard solution by using high-resolution mass spectrometry with Q-exactive. On the bottom, you can see extracted peak by MOVZ 212 9792 representing PFBA. We clearly have PFBA in our background from blink injection. We did many following-up experiments to determine where this contamination was coming from, and we traced it to leaching from our old LC system, probably from some flora-containing seals. When we switched to another LC systems, we did not observe this problem anymore. The other problem that we encountered was observing a high background for PFPA for MRM transition 263 to 19. When we look more close into it, this is transition 263 to 19 for PFPA at the lowest calibration level by triple quadrupole. At the next calibration level, 0.1, we do see a pronounced peak, but it's probably no more than 3 times above noise level. Finally, only at our middle calibration point, we do see the defined peak. Now, if we do the same by using high-resolution mass spectrometry for 262, 97, 60 MOVZ, we see pronounced peaks at all low calibration levels. Now back to our PFAS puzzle, let's look into extraction, our first sample preparation approach. We have developed and validated extraction protocol for food packaging. We tested different solvents and extraction conditions for both incurred and spiked food packaging. I'm not showing the results of this method development here. We have published it in our paper. If you want more information, please refer to our published paper or email me if you want a copy. But in short, our extraction protocol entitled taking 1 gram of food packaging samples, adding internal standards, shaking and sonicating it with 6 milliliters of methanol by shaking for 1 hour and sonicating at 45 C for another hour, centrifuging, collecting supernatant, evaporating it down with nitrogen, and reconstituting it with 1 milliliter of methanol. Here's a paper that I mentioned that we published. Happy to share with you if you're interested. Methanolic extracts of food packaging samples were analyzed with both targeted and non-targeted approaches. I will present the data on targeted approach, and as I mentioned, Kevin will present data on non-targeted approach. However, what I want to show here is by suspect screening using non-targeted approach and two lists, APA PFAS master list and NIST PFAS list, we were able to find 6,2-fluorotelomere phosphate ester, otherwise known in 6,2-DPAP, in our samples. We purchased analytical standard and confirmed its identity, and we added it to our targeted list so we could analyze for that as well, and we were also able to get isotopically labeled internal standard. These are the results of what was found in methanolic extracts separated by food packaging types. On the x-axis, we have sample numbers for each category. On the y-axis, we have concentrations of PFAS measured in nanogram per gram. And here in the box, you can see the list of detected PFAS analytes. 19 of 34 were detected overall. The take-home message here is 84% samples had detectable levels of at least one PFAS, and the PFAS that we found by suspect screening, 6,2-DPAP, was the most frequently detected compound in 75% of samples and also at highest levels. Interesting information about PAPs and DPAPs. Polyfluoroalkyl phosphates, PAPs, are common non-polymeric surfactants used in mixtures of fluorochemicals for oil and grease resistance in food contact materials, and industrial PAP mixtures contain mostly DPAPs. Similar to our findings, 6,2-DPAP was the most frequently detected PFAS analyte and paperboard grease-proof food packaging samples from Spain. A recent study testing PFAS in fast food packaging from France also found 6,2-DPAP in every single sample they analyzed. And finally, it is not just in food packaging. This recent paper also found 6,2-DPAP by suspect screening in similar way we did in cosmetics and personal care products, and it had highest frequency of detection and highest levels compared to other PFAS found in this study. If we look into geographical origin, the highest levels were measured in samples from North America and low level in samples from Europe and Asia. Another interesting point I want to make is the different composition patterns for PFAS. With the exception of 6,2-DPAP, it appears that substances found in packaging from Asia contain 10 to 14 carbons, while PFAS substances detected in European food packaging contained 8 to 10 carbons. This finding is consistent with previous report from 2017 testing popcorn bags from different geographical locations. They found that short-chain PFAS C4 to C8 were more common in European and American countries, and long-chain over C8 PFAS were more common in bags from Asia, probably reflecting the usage pattern in different geographical origins. Now that we covered extraction results, let's look into the next puzzle piece, total oxidizable precursor assay, or TOP for short. The same methanolic extracts of food packaging samples was subjected to TOP assay. As the name suggests, TOP uses oxidation to convert oxidizable PFAS precursors into the terminal degradation products. Here's an example of PFAS precursors and their oxidation products. In our work, we choose 3 PFAS precursors, NITFOSA, 6,2-FTS, and 5,3-FTCA, to conduct experiments on method development to determine oxidation rates and look into recoveries of fluorine after oxidation. TOP assay was first developed by Hautz and Sedlak in 2012 for urban runoff, and this paper from 2017 utilized TOP for papers and textiles, and this is what we used as a baseline to develop and validate our TOP method. The basic steps of top protocol including evaporating methanolic extract, adding potassium persulfate and sodium hydroxide, and incubating at 85 C for 6 hours. After adjusting pH, methanol is added to vortex ultracentrifuge, and supernatant is transferred for LC-MS analysis of terminal products. This graph presents results of our top assay for food packaging samples. Overall, 8 terminal products consisting of 5 carboxylates and 3 sulfonates were found. On the y-axis, you see summed levels of these 8 found compounds in nanogram per gram. And what you can see is that the levels increased over 10 times from before top assay to after the oxidation, which suggests that there are some PFAS precursors in food packaging samples that they're not captured by our targeted analysis covering 34 PFAS we selected. Similar to our results, an order of magnitude increase after top assay was observed for PFAS products in pet food packaging samples published in this paper. Another recent paper by the US EPA scientists highlighted benefits and challenges of top assay and the need for standardization of this technique. Based on both extraction results and top assay results, 25 samples were selected for migration tests. We selected the samples that contain the highest levels of PFAS and also where PFAS were found at highest frequency of detection. Migration tests were conducted under controlled migration conditions as listed in this guidance in the bottom of this slide, U.S. Food and Drug Administration guidance for industry. Based on the food types, we selected 2 different food simulants, 10% ethanol, 90% water, and 95% ethanol, 5% water. Samples were incubated at 2 different temperatures, 20 C and 40 degrees C, representing storage conditions at refrigeration, freezing, and shelf conditions. And finally, the migration test was conducted by complete immersion, where 1 square inch or 2.5 by 2.5 centimeter of food packaging material was immersed in 10 milliliters of food simulant in polypropylene tubes. Each sample was tested in 3 replicates and also included blinks. The blinks were just food simulants with no food packaging material in it. In order to study the dynamics of migration, aliquots were collected from each sample at time of 2, 24, 96, and 240 hours, that is 10 days. And also each second replicate of each sample was spiked with isotopically labeled internal standards for quality assurance, quality control, and also to guide in semi-quantitation or quantitation of PFAS that were later found using non-targeted approach. We found 4 PFAS analytes in 5 food packaging samples. This graph shows 2 PFAS found in salami sample consisting of plastic and paper and tested with fatty food simulant representing fatty food, 95% ethanol at 20 degrees C representing refrigeration. On y-axis you can see migration time in hours, and on x-axis concentration in nanogram per gram. PFHXA in orange dots and PFHPA in blue dots shown here were found in the sample, and if you look at the dynamic of migration, you can see that with prolonged interaction of food packaging with food simulants, the levels were increasing. The second sample was cake food packaging consisting of paper, again 95% ethanol food simulant tested at 40 degrees C representing shelf storage. PFHXS was found and at about approximately the same levels, showing similar dynamics. We found 6,2-D pop in lamb kebab samples consisting of plastic and tested with 95% ethanol food simulant at 20 degrees C. Here on this graph, you can see a rapid transfer of 6,2-D PFAS from food packaging samples into food simulant. And rapid transfer happened at about 2 hours. And you can see this dynamic of migration is different from what we observed for other PFAS. 6,2-D PFAS was also found in cookie sample, and that was coated paper tested with 95% ethanol at 40 degrees C. What I also want to point out that for 3 out of 4 migration samples, the levels were low. For salami, cake, and cookies, the levels were below 1 nanogram per gram. However, the levels of 6,2-D PFAS found in lamb kebab samples were at 10 nanogram per gram. Finally, the 5th sample where we found PFHXA migrating was tomato sample produced consisting of foam and film tested with food simulant of 10% ethanol at 20 degrees C. However, the levels were so low, just slightly above our detection limits, that we could not observe dynamics of migration. Now, what does it all mean? The ultimate goal of our study was to estimate the exposure, and for this we calculated dietary exposure estimates. I have to say that there are currently no regulations in the United States for food or food packaging for PFAS. We do have regulations set up by US EPA for PFAS in drinking water. However, the European Union, European Food Safety Authority, came up with regulations for certain PFAS in certain foodstuff in December of 2022. EFSA suggested to use tolerable weekly intake TWI of 4.4 nanogram per kilogram body weight per week for the sum of 4 PFAS: PFOS, PFOA, PFNA, and PFHSX. We calculated cumulative estimated daily intake, CEDI, for PFAS that we measured in our migration samples, and we took the highest levels found at 240 hours of migration. CEDI was calculated using food type distribution for each food simulant and packaging category and consumption factors for packaging category, and these numbers were found in this guidance document. Furthermore, we calculated weekly intake by multiplying CEDI by daily consumed amount of certain food in kilogram and dividing it by average body weight of 70 kilogram and multiplying it by 7 days. The problem was there are 4 PFAS that are regulated: PFOS, PFOA, PFNA, and PFHSX. However, we found 4 PFAS, which is PFHSX, PFHAA, PFHPA, and 6,2-D PFAS, so only one was overlapping. In order to solve this problem, we use relative potency factors to convert PFAS which are not regulated into PFOA equivalents, and we use these numbers from this publication. This table shows what we found, what PFAS we found, the relative potency factors for conversion to PFOA, and proposed daily consumed amount in kilogram. So what I want to point out, for example, that for cake and cookies, we selected 0.2 kilograms, that is 200 gram consumed amount per day, or for those of us in the United States, that's about 7 ounces. So that's a very generous amount of cake and cookies to eat every day. And for salami, we selected 0.5, which is half a kilogram or about 1 pound. So the culmination of our efforts is calculated weekly intake here for 5 samples that we found PFAS in, and on y-axis is nanogram per kilogram body weight per week. So the levels that we found, even if we sum them all, were all below EFSA regulated values of 4.4 nanogram per gram. So yes, you can have your cake with PFHXS and eat it too. However, what I want to point out is there are only 4 PFAS that are currently regulated, and more alternative new and emerging PFAS are on the market, of which we don't know the identity and we don't know at what amounts they're being applied and how toxic they are, the most important thing. Also, for these calculations, we use 70 kilogram body weight, but children, for example, who consume this cake or cookies do not weigh 70 kilograms. And we have to rely on another piece of puzzle, non-targeted analysis, in order to capture this PFAS analytes that we are not aware of, that also can be contributing to high levels of oxidizable levels that we measured after top assay. Because what we measure by targeted analysis is only a tip of the iceberg, and non-targeted analysis is extremely important to capture analytes that are missed during targeted analysis. Which brings me to the end of my presentation, and with this I would like to acknowledge our funding source, USDA National Institute of Food and Agriculture, NIFA, and the grant that we received from this agency for this study. I would like to acknowledge my collaborators with University of Pittsburgh, Dr. Carla Eng and Mega Bedi, and my colleagues in USDA ARS helping with this work. And with this, I would like to thank you for your attention, and I will be happy to take any questions you may have. Thank you.